السلام عليكم ورحمة الله وبركاته، ومرحبا بكم جميعا في احتفالنا بيوم العلم. قبل ما نبدأ ماذا بيا نشوفوا شكون راوي يشارك معنا من أي بلد أو من أي ولاية. حطوها في الشات، يعني البلد أو الولاية اللي راكم تشاهدوا فينا منها. قبل ما نبدأ أود أن أشكر كل اللي فولونتير، يعني the scientists اللي كانوا العلماء الجزائريين اللي malgré كما يقولوا très occupés, they are very busy، مشغولين بزاف في مجالهم المهني، ومالغري صح حبوا يشاركونا يعني comme des volontaires باش نحتفلوا بيوم العلم في الجزائر، وهو احتفال بالعلامة ابن باديس، وهو من العلماء الجزائريين المؤسسين للحضارة الجديدة في العلم في الجزائر وجمعية العلماء المسلمين. شكرا للدكتور أحمد شنة، شكرا للدكتور محمد بوجلال، فايز نايت محمد، دكتور بلحسين، دكتورة سيهم بودينا، وكذلك الدكتور نبيل أوفن على المشاركة. في عنا برنامج طري لمدة 3 ساعات. البرنامج فيه 4 محاضرات من بلدان عبر العالم، من الإمارات، من قطر، من الولايات المتحدة والجزائر. إذا كان عندكم يعني أسئلة حطوا في الشات question ماجسكول، وبعدين حطوا السؤال تاعكم، وراح يكون عندنا 30 دقيقة اللي هي بريزنتاسيون، وبعدها يكونوا 10 دقائق للأسئلة والأجوبة. ولما جاوبناش على الأسئلة تاعكم we are tracking them، وبعدين اللي سجلوا في المحاضرة راح نبعثوا لهم يعني الأجوبة عبر الإيميل إن شاء الله. وبهذا نبتدئ بمشيئة الله مع الدكتور أحمد شنة. نجيب الدكتور أحمد شنة معايا هنا on stage على بركة الله.
I would like to ونحب نشكر المنظمين لهذه الإيفنت، وكالعادة يعني أصبحت يعني عادة كل سنة يعني نحضروا هذا الاجتماع أو هذا يعني السيمينارز كل سنة. حاب أولا نقدم العلامة يعني دكتور سعيد درميم، هو غني عن التعريف وخاصة في ميدان تاعو في الإميونولوجي. دكتور سعيد تحصل على الدكتوراه في جامعة، أولا درس في قسنطينة، ذن تحصل على منحة يعني ذهب إلى بريطانيا، يعني درس في جامعة سالفورد، عمل ماستر ثم PhD، انهى PhD تاعو immunology 1992، ثم اشتغل يعني في NCI في ميلان as a postdoc، ثم National Institute of Health، ثم يعني he went and led the lymphoma cancer vaccine team at the Peterson Institute for Cancer Research in Manchester for 5 years, and he also published over 100 papers. He is also reviewer to many journals, and he's associate editor for Frontier in Immunology, and reviewer to multiple prestigious international journal. Dr. Saeed currently holds a position as a senior scientist and director of translational cancer research in Qatar. I think Dr. Saeed, I think I cannot. He has so many achievements and participation in different conferences, and you can read all his work in over 100 papers. So I don't want to, I cannot talk enough about Dr. Saeed. So he can now present to you his presentation about CAR cells in hematology malignancy. تفضل دكتور سعيد.
Thank you very much, my friend Ahmed. I will be very, very honored to present to you this afternoon and share with you this presentation. The subject will be on CAR cells therapy which is a new era in cancer immunotherapy. Just for the picture I'm sharing with you now, on my right hand is the center where I am working, and on the left hand is the new machine for the car cells generation, which is called Prodigy. This is in my facility. We are just started the preparation and getting this technology in our facility. So why we want CAR T-cell treatment, why we want to develop this? I will share with you the CAR structure and also the mechanism of this CAR T-cell killing of the cancer. I will talk about how CAR T-cell therapy works, and I will focus on the therapy in hematologic malignancy. Of course, CAR T-cells is as well being developed in solid tumor, but I will focus in the hematologic malignancy, which is well developed. I will talk about the CAR T-cell targeted antigens in hematologic malignancy. I will share with you the FDA-approved CAR T-cell therapy, and as well as the clinical outcome of CAR T-cell therapy. Later on, I will share with you just a review that we recently published talking about the development and the bad, the good, and ugly of CAR T-cells in solid tumor. Then, if you are interested more, you can go and look at this review. So I will share with you the basic cellular and humoral immunity against cancer cells, just for the people who doesn't have a background immunology. For the cellular immunity, cancers can process a tumor antigen and present it to the T cells through the major histocompatibility class 1 or class 2. The T cells can get activated, then they release granzyme and perforin and result in the killing of the T cells. For the humoral immunity, sorry, I did animation. Unfortunately, you don't see, but I can just summarize for you this. For the humoral immunity, the B cells will recognize the whole antigen present by cancer cells and get activated and result in B cells. B cells will release antibodies specific to these antigens presented by the cancer cells. And the result for this antibody, they will kill cancer cells through three different compartments. Let me just go back. Yes. The first one is antibody-dependent cytotoxicity T cells, which is initiated by the natural killer cells. The second one is antibody-dependent cellular phagocytosis, and this through the macrophages. And the last one is the complement-dependent cytotoxicity, which initiated by the complement pathway. The expected object is the cancer death of these cells. Unfortunately, cancer can escape the immune system through different mechanisms. One of them is defect in major histocompatibility class 1 production, which can render cancer cell invisible to the cytotoxic cells. And another mechanism is immune suppression through the tumor cell and tumor microenvironment. And And for this, recently they have developed several immunotherapy treatment or potential treatment. One of them is antibody that inhibit the immune checkpoint inhibitors, which is antibody against the PD-L1 and anti-PD-1. I will not talk about that. This is another subject, but I will focus on the last one, which is they develop CAR T cells to overcome such immune escape. CAR T cells are T cells equipped with chimeric antigen receptor, which is genetically engineered receptor that combine the specific binding domain from tumor-specific antibody, such as CD19 present on B cells. I will talk later in details about that. So this that target specific tumor-associated antigen with no need for classical antigen processing and presentation by the MHC. So as I mentioned before, they will make single chain antibody that is recognizing the whole antigen present on the cancer cells, and they can attach this to an intracellular domain of the CD3. This is the one, the CD3, the intracellular domain, and they result in both high affinity and specificity for the antibody and high potential for cytotoxic and proliferation through the signaling of the CD3 domain. So in that case, CAR T cells will share both advantages of a humoral immunity and cellular immunity. So it's a weapon with two faces. The cellular part through the CD3 signaling, and the other part is the recognition of the single chain antibody of the whole antigen. CAR T cell can specifically recognize the tumor-associated antigen. They exercise their cytotoxic activity by releasing, as I mentioned before, perforin and granzyme, and by activation of dead cell receptor pathways such as Fas and Fas ligands, and this will lead to cancer cell apoptosis and necrosis. Activated CAR T cell also secrete interferon gamma and TNF alpha, which can activate natural killer cells and result in anti-tumor cytotoxicity. For the last decade, they developed CAR T cells with different generation, the first generation to the next generation. The first generation will contain, as I mentioned before, the SSV for the antibody recognition that is linked to a signal one, which is the intracellular domain of CD3. But this one, unfortunately, didn't work perfect and didn't maintain the cytotoxicity activity of the tumor, and the tumor will come back to the patient. Therefore, they moved to the second generation, which they introduce, in addition to a signal one of the CD3 domain, they add a signal 2, which is a co-stimulatory signal, either CD28 or CD134, CD137, and this is the most successful one. I will focus in my next presentation on the FDA approved, because most of the one that approved is through the signal, the next generation that have signal 1 and signal 2. Of course, there are other, the last three when they are being developed, where they have they either introduce another co-stimulatory, third co-stimulatory signal, and for example, the fourth generation, they introduce interleukin 12 as well to produce in the tumor microenvironment, and this will result in the expansion of the T cell and activation for the T cells. And the last one, the next generation, which is not in the market yet, is the introduce as well transduction signal through the JAK and STAT3, STAT5. How CAR T cell therapy works? So CAR T-cell therapy is a complex multi-step process for patients. First of all, we need to collect T-cells, then engineer and expand them, then cryopreserve them, give conditioning therapy to the patient in a similar way that is given to bone marrow transplantation and stem cell transplantation. Then you can infuse the CAR T-cells, and of course, you have to follow up on the patient. I will take you step by step in this presentation. So for the T-cell collection, T-cells are collected from patients via leukapheresis, a process that withdraws blood from the body and moves through a cell separator to collect T-cells. The remaining blood compounds are then returned into the body, and you can do that in 6 hours in a hospital. So for the T-cell engineering and expansion, the T-cell are sent to a GMP cell therapy facility where they are genetically engineered to target specific type of cancer. Then they expand these modified T-cells through this prodigy machine, the one I share with you at the beginning that we have already in the lab. And this is the process. Is automation. You just put the bag and you put the vector, and they will transfect or infect the cells. Then later the cells will be expanded. So T-cells are activated, provided with a CAR molecule, which is using viral vectors such as retrovirus and lentivirus, or non-viral gene editing such as the new developed CRISPR-Cas9. Of course, they use retrovirus and lentivirus because these two vectors, they can infect cells even if they are not dividing, which is a potential way to infect the T cells because they are not proliferating. The most common and effective method for gene transduction in CAR T cells production is using viral vectors, and the most approved now, FDA approved, use this technology. After engineering and expansion, the CAR T cells are cryopreserved in liquid nitrogen in the vapor phase. At the same time, patients may receive chemotherapy for their cancer conditioning therapy. I mentioned it before. This helps to create a system for the infusion of CAR T cells, because this is the way that you have to get rid of the other one to replace the new one. Then the conditioning therapy. The CAR T cells are thawed before we give to the patient and infused through a central line in a process similar to the blood transfusion. The patient may receive medication to prevent and control possible side effects of CAR T cells. I will mention this at the end about the side effect and what type of drugs they will give as well to the patient. CAR T cells that have been returned to the patient bloodstream are attracted to the target on the surface of cancer cell. They proliferate, then they identify the cancer cells with target antigen, then they will kill the target, and some of them, they will become memory and maintain in the patient just in case if the cancer recur again. Patients who receive CAR T cell therapy have a risk recovery period of approximately 2 to 3 months. During this period, patients will be evaluated for treatment response and for side effects related to CAR T cell therapy, such as this toxicity, such as tumor lysis syndrome, cytokine release syndrome, neurotoxicity, on target of tumor toxicity. I will discuss this in detail. So the cytokine release syndrome manifesting where excessive release of cytokine is triggered by CAR T cell activation, proliferation, and enhanced killing, associated with a broad range of clinical symptoms. The toxicity management, fortunately, they have a monoclonal antibody against the IL-6 receptor, and this acting as an immunosuppressor and is used for the treatment of CRS, effective treatment for this toxicity. The second type of toxicity is ICANS, and for the management for this toxicity, they use corticosteroid and is effective as well. The tumor lysis syndrome, the management should therefore include prevention of cardiac toxicity as well as prevention of renal function. Then on targets of tumor toxicity, you remember, for example, if we are using the CD19, which is present on B cells or malignant B cells, this antigen as well is not specific and is present on B cells. And to overcome this toxicity, researchers are working hard to find neoantigen, also called tumor-specific antigen. They are looking for antigen that is already exclusively expressed on the cancer cells and not in other organ or normal cells. So for a successful CAR T-cell therapy, they have to have a maintained CAR cells, a high percentage of maintaining of CAR cells in the patient. They have as well to have a high interferon secretion in the blood, and they should have very low minimal residual cancer or minimal residual disease, and they should have a high overall survival, ORR, objective response rate, complete response, and duration of response. This is a successful CAR T cells. I will share with you two examples of the first development of CAR T cells. This was in 2016. They used 7 patients with refractory diffuse large B cell have received CD19 CAR T cell targeting CD19 molecule on the lymphoma cell, while some experienced significant therapy-related side effects or recovered fully, with several patients achieving a huge tumor shrinkage, as demonstrated in this slide. These patients received 2 million CAR T cells per kilo, and after 4 weeks or a month, as you see the difference, there is a disappearance almost completely of the tumor. The second example is a 62-year-old male with refractory large B-cell lymphoma who had previously metastasis. You see in the first baseline all over the body and the lungs, and after a treatment, the patient after 3 months recovered from the disease and maintained after 18 months disease-free. So what are the main cancer marker that is being used for different hematologic malignancy? We have the CD19, which is a B-cell activation and proliferation marker, is highly expressed in most malignant B-cell tumor. CD20 as well, highly expressed in B malignant cell and could regulate cell activation and proliferation. CD22 expressed in most B-cell lymphoma, in B-cell as well in leukemias. The tumor antigen on T-cell tumor targets CD5 and CD7, and multiple myeloma we have the BCMA and CD138. So till today, 1,253 clinical trials have been carried out worldwide, in which 75% were in hematologic malignancy, 70% were targeting the CD19. So out of 945 clinical trials, only 6 CAR T-cell therapy are FDA approved. Let me go back, and I'll share with you an example One of them is the Chimera, produced by Novartis, 2017, CD19 second generation. You remember, and it was in children and young adults in refractory or relapse B acute lymphocytic leukemia. The overall objective response was 81%, complete response was 66%, and they have a toxicity with 46% of the CRS and 30% of ICANS. Unfortunately, I cannot see my slide here to continue telling you the rest of the generation. This was animation, but I cannot see them. I don't know if the technical people will help with this slide. This slide is very important, but I can just summarize if they cannot help technically. I'll just wait.
Yeah.
Yes,
Was
this
that,
is a very important slide. I need to see the rest of the.
is that open with the animation?
Yes, yes. I
Yeah.
don't know why you cannot use the animation in the. You should have access to the animation.
Yeah, I didn't see it too. It's unfortunate, but we'll have to go with what we have.
Yeah, yeah. In that case, I have just to mention in this one, they are from 2017 till 2020. The last one, the last drug used gave a huge, more than 90% of the objective response, and I think about over 70 of the complete response. And importantly, the toxicity was very low for both CRS and ICANS, and this one was carried out refractory relapse multiple myeloma. This and is not against CD19, but is against the myeloma antigen that is expressed in myeloma antigen, and this one is being used now as exclusively a good drug to treat an multiple myeloma. Of course, this technology has advantage and disadvantage. The advantage is it is powerful, that attacks systematically and deeply, offer durable protection, recognize antigen in HLA independent manner, which is very good because of the escape of the immune response through downregulation of HLA that cancer mainly use, target different type of antigens such as protein, carbohydrate, or glycolipid, it doesn't matter what type, and have generated impressive results in the treatment of patients with hematologic malignancy, as I mentioned. The disadvantage, as I mentioned, the cytokine release syndrome, neurotoxicity, and tumor lysis syndrome, but again, an on target of tumor cytotoxicity, but scientists have developed already treatment for this and it's been used. And loss of expression, as I mentioned, of tumor-associated antigen. And finally, the most important disadvantage is the high cost. For example, the drug that's provided by Novartis, it costs $475,000 US per patient. Anyway, CAR T-cell are now proven treatment approach for hematologic malignancy, but challenges remain around T-cell efficacy, safety, long-term persistence, and ability to target solid tumor. And this is the one recent review that we summarize all to do with the CAR T-cell in solid tumor. You can have access to this, is provided free of charge online. And thank you very much for your attention. And I am very sorry that I couldn't present most of the data because it was animation. I didn't know that you cannot use animation in this technology of presentation. Thank you very much.
Thank you, Dr. Saeed, for this nice presentation, this overview about this technology that has been for a few years and mostly applied now in the West. You know, unfortunately, the developing country, they don't have this technology and it's very expensive for them. So I have one question first, you know. So I've been following these for many years. So, I mean, I know that the leaders looks like that they're like BMS, BioNTech, and the CARE group. They have the highest number of patents in the field. How you are practicing it in Qatar, for example, and so how you are practicing it? You are practicing this, I think you are treating some patient with this technology.
Yes, we are planning. We didn't get technology. I just received the technology, I think a year ago. We are working. We did already send people abroad to be certified for the production. We need to get we got the plasmid for the CD19, and we're going to do some of the standardization in the lab. Then we have to do our own probably phase 1, phase 2, even if it is approved just for our lab, and we can move for this one. It will take time to do that, but I know in the region, in Saudi Arabia, in King Faisal Hospital, they already treat, probably the only place in the region that they treat 50 patients with CAR T cells that is produced outside. But now they are in the process of producing. We're trying to set up a collaboration with them to do a technology transfer.
So which technology, which part you are collaborating?
We'll do, yeah, the CD19 for the treatment of hematologic malignancies with CD19 expression marker.
Okay, thank you. Okay, let's see if any questions from the people.
Can I answer this one, the first one? Yes?
Yeah, I think...
I see a question. Does the CAR T-cell therapy have the same effectiveness in solid tumor as in hematologic malignancy? The answer is no. This is why it's not been FDA approved yet. Because the CAR T-cells for solid tumor is very difficult to get to home to the tumor because it's a solid. Whereas in hematologic malignancy, when you do the infusion, you will get it in contact with leukemia or lymphoma. So this is, they are developing now, putting homing marker for the CAR T cells to home to the tumor site.
There is a question here from Ahmed Marden. Said,
what? Yeah. Can I see the question again?
Do you want me to? It is in the.
I think he's talking about the preferred vector. It's the lentivirus. Lentivirus, yeah.
No. This question is different. She said.
This is another one.
Yeah, this method. This method is efficient in what clinical stages level, or it can be used even after metastatic stage, which is the most hopeless one.
Yeah, I showed the case at the beginning where the patient has a huge metastasis and recovered. Yes, it can be used as well. Actually, those are used for time being for the patient who is in at advanced stage.
Yeah, this one, I don't know if you answered or no. Salma Shami asked, does this CAR cell therapy have the same effectiveness in solid tumor as in hematological malignancy?
Yeah, I already answered that. It won't have the same effectiveness in solid tumor because of the homing, the problem of homing. The T cells difficult to home to the organs, but for hematology, they have access already in the peripheral blood or even bone marrow.
Okay. Another question from Arab Iman. We do find a bit of problems with the transduction of immune cells. Do the T cells also exhibit same decreased transduction efficacy during modification step?
Yeah, the T cells, because they are not providing. You have to use an antivirus, as I mentioned, that transfect the cells, and you have to have the right vector. Once you use the virus, it will integrate in the cells that you need to expand the cells to maintain them. I don't know exactly what she meant by the difficulty in the transduction. You should not have any difficulty when you use antivirus. If you use other methods, mechanical methods, of course, or use lipids-based transduction, you will have no efficient transduction.
She's saying about decrease in efficacy during modification.
Yeah, because you need to make sure that that is efficient at the beginning. You have to have, let's say, the transduction 90% or something. Then this one will maintain. If it is a lower transduction, then you will expand non-transduced cells, and they will overgrow over the transduced one. So you end up with less cells.
Okay. Shamsuddin asked, thank you for the presentation. In the trial, CAR T cells are always associated with the chemotherapy or can be applied alone?
I don't know what he means, but associated with chemotherapy. But usually,
in addition, maybe in addition.
Yes. In addition, usually the patient will have other treatment, and if they fail all those and the tumor recover, they will use the CAR T cells. But I think in the future now, they are planning to use it from at the first stage. So you will have more effectiveness because the patient is already stressed with all the other treatments. So if you use CAR T cells at late stage, it may not have as good as effective, will be effective as if you use it from first stage.
Okay. Another question, can the effectiveness of CAR T cell therapy be improved by combining it with other immunotherapy agent or targeted therapy?
Yes, there are many clinical trials now. They're using the combined CAR T cells with different therapies such as immunotherapy. And I will recommend to read my review. My recent review is all there. All the combination is mentioned in the review.
Maybe this is the last question because we have to move on. Santosh asked, is there any other technology other than I don't know,
Mitini Prodigy, the Prodigy machine.
Yeah, what is your feedback on thermogenesis?
We don't use that, so I have no comment on the other technology.
Okay, last maybe, how long do the effects of CAR T cells therapy last, and what is the future of this technology, research and development?
How long it will stay? It will stay a long time for in the patient. Some patients are living with it now, completely cured. Of course, they are developing more for the main thing is not to sustain the T cell, but to have an effective, a quick effective on the tumor killing, like the next generation I mentioned in the last when they introduced some signal transduction, or in the fourth generation where they introduce an interleukin 12 that is known to be released in the environment and enhance the T cells to not just to have the CAR T cells clearing the tumor, but to activate as well natural killer cells to help as well in clearing of the tumor.
Okay, this is my last question. The bottom line for the developing world, how to reduce the price for this expensive technology and how to make it accessible to the people in Africa and Asia, for example, and South America.
Yeah, I think we have to go through in-house production. In-house production, because you can buy the vector. Is cheap if you buy the vector. The cost before is the manufacture. You will send them the bag of PBMC from the patient, and they'll give you a bag ready of CAR T cells. This is why the cost. But now if we have the vector, we can produce that, do the quality control, everything, and treat the patient. That's we reduce. I remember I was talking to a company that they are ready to help in that with the vector. It may cost even $10,000 or something like that, just for the vector to have a license to use it.
I mean, we know that lots of expensive drugs, for example, like for HCV, for example, it costs $65,000 to treat a patient in the US, but they allowed it to be made in India, and they can distribute it to about 5 to 10 poor countries, and the price went down from 65,000 to 1,000. Is there any way also to take it to this technology to India, for example, and they can provide it to a developing country like other drugs?
Yes, even in our countries, we just have access to technology. You get the vector, and the rest is just production. The machine for the Prodigy will cost, I bought my machine at the cost, I think, 1.2 million riyal, which is, I think, over just, $400,000 or $300,000. So it's nothing compared for other equipment. Then the matter is the expertise. You get the expertise who can produce this one and run the technology, do the quality control. The management of CAR T cells is easy now after COVID-19, because COVID-19 caused as well the cytokine syndrome, and which is the release of a lot of cytokine, and people during that time they treat with antibody to IL-6 and interleukin-6. Now this one technology is available in most hospitals to treat the follow-up of the patient, the toxicity. The other side, now there are clinical trials that they prove they can reduce the cytotoxicity. So that one, as I mentioned, for example, for the multiple myeloma, now we are talking about 3 and 4% cytotoxicities for the neurocytotoxicity. I mean, that's great. I mean, it's a big achievement for that. So I think the future of CAR T cells is in the reduction of the cost and making in-house. You could, Ahmed, imagine many years ago, bone marrow transplantation or stem cell transplantation is done maybe just in USA. You have to send the patient to there or in Europe. Now in small cities, for example, in Boudina, in Algeria, there is a bone marrow transplantation now, not just in Algiers. So I think it's just a country should focus on technology transfer. This is the way to reduce the cost and have this treatment accessed by many patients.
Thank you, Dr. Saeed, for this nice presentation. Really, it's a really good education for all of us. So thank you, and for your contribution as usual. Yeah. So now we are going to move to another talk, which the title is, the speaker is Mohamed, Dr. Mohamed Belhocine. The talk is going to be Personalized Medicine, a New Treatment Paradigm. Dr. Belhocine, he's a well-known expert in bioinformatics. He's a senior bioinformatician at the Molecular Biology and Genomic Center in Dubai, has expertise in both experimental and computational biology. He focused on his work on large-scale genomic approach to understand genetic and epigenetic mechanisms associated with normal and pathologic cell development. He supervised different projects in the clinical trial for next-generation sequencing and data analysis. Dr. Belhocine developed and oversee research and training at various universities in different locations. So without delaying, I'd like to ask Dr. Belhocine to start his presentation. Thank you.
Assalamu alaikum. You can hear me?
Yes, we can hear you, yeah.
Okay, it's better the sound now. Okay.
Yeah.
Good. Thank you for the kind words and presentation. And thank you for the organizer for this Day of Science, يوم العلم اللي نذكروا فيه كلنا ابن باديس وحبه للعلم والجزائر. من غير إطالة راح نبدأ البريزنتاسيون تاعي where I will speak about the new paradigm of personalized medicine and give in the end small practical case how we can use this new technologies, new medicine to treat patients or to help to improve lives. So the term personalized medicine, genesis, start reality 2 millennia before, when Hippocrates was making like combining to assess make assessment for a patient by combining the 4 humor, blood, phlegm, yellow bill, and black bill. So the personalized medicine, it's not something really new, but what is the new in this day? It's we have now the tools and the technology to make it really personalized and to enter a new era of the medicine. For me also the animation, it's not working. Okay. Anyway, so there is the genesis of this new era start by 3 problems in reality. The first problem, it's the patient intrinsic genetic, between parentheses, genetic variability. It will cause that if we give the same medicine for the the population, not all of them will benefit from this medicine because of their genetic background. And we can see this with different drugs in different area. So this is was the first observation that we made, that if not every human will benefit from the same from the drug with the same way. The second problem was the dosage. It's not one size fit everyone. So there is the problem to find the correct dosage and for each drug to the individuals. And the third problem was to find the right drug. So there is around 40% of diseases, they have a mutation or a problem. They can be treated with a specific drug if we know that it's the case. So the problem here, we cannot see the animation also. It's covered. It should be this removed. Okay. So the traditional medicine, it was to find, you have a group of patients with the same disease or same problem. We give them all the same therapy, but the group of one group will benefit from this drug, but not another group, no. And even there is third group where they will be a problem, not benefit, but they can get some problem. But the new personalized medicine that you cannot see the presentation here, it's to use the new technology or the new diagnostic tools to identify the variation in the population and to adjust the treatment and give the right drug to the right people and to get the higher benefit from the medicine. So to summarize what is personalized medicine, we can call it precise or individualized medicine also sometimes. And it's to use the diagnostic tools to identify specific biological marker, oftenly it's genetic, to access some medicine and treatment and to give them the best of this treatment. Okay, here's the same thing. So as I said before, it's not a new concept, the personalized medicine, but what is new, it's the tools and the technology that we reach and that can help us to now open this door of personalized medicine. So the old medicine will use all this new technology like the mobile connected device, the new imagery system, the new scanner, ARM, all this technology, and plus the genomic and the era of the genetic analysis to bring us to the new medicine. So it's like it's détruire l'ancienne médecine and to regrouper la nouvelle médecine dans une nouvelle personalized medicine within all these new tools. So, for example, for the new technology, you have like around 30% of the population are using or wearing monitoring systems like this Apple Watch, where they monitor the heartbeat, pressure, blood pressure. That all this information can help the doctors to have some information and to understand the disease or what is the problem. And also we are entering the new era of bionic human, where the human will be connected to some devices like this one. I am already wearing one of like this, and this it's to monitor the sugar in the blood, the glycemia, and I am sure in the near futures we will have other devices monitoring other parameters in our body and give the doctors a better view of our health. And also the genetic, like I said, and genomic, with the explosion of different projects in the world, like these are different projects, genomic project, where they are sequencing thousands of millions of samples of genomic to create databases of variation, SNPs, deletion, to find what is the polymorphic and separate polymorphic from the variant that they are pathogenic, or even the variant that they give more help to not pathogenic, but help the patient to get better use of medicine or drug. So, and here I summarize like a small picture of what is new in this new era, not only genomic, but proteomic, single cell sequencing, epigenetic, all these devices connected. And with the era of AI and the big data, all this can be collected, and we can like give a better view of the health and monitor better the health of the patient. But I put this comic image here to show that also it's good, but it's also difficult now with all this data. We need to have tools how to find this needle inside in all this the information. So there is Isaac Asimov said one time, science gathering knowledge faster than society gathering wisdom in this time. I think we are in this era that all this information we are collecting. We need now to sit and to start looking and to organize and all this information and to use them to improve our health system. We cannot see in reality here. I put some important date for medicine, personalized medicine, but the problem of the animation. Okay, just to say that during the first breakpoint was the human genome, human sequencing in 2000, and this creates like a big jump in our comprehension of the mechanism and the phenotype, how the phenotype are controlled by all the pathways, the gene expression, the epigenetic, and all this. Sorry, I cannot see well. Okay, I will pass this one. So the quiz, what is the first personalized medicine we used? So the first one was the Herceptin. It was improved in 1998 to treat breast cancer with this mutation positive. And here FDA announced that it's a new era for treatment for medicine, personalized medicine, since we will not consider all the cancers like one disease, but it's a single disease. Each one of them are a single disease and need to be treated or like viewed like independent from the other one. So with the genomic development and the bioinformatics especially, so we passed from 16 years ago, we passed from classifying cancer from a disease of a blood to today, where we have more than 40 unique different kind of leukemia or 50 unique kind of lymphoma, where each one of them can be treated with different and specific drug. So the genomic and bioinformatics really bring in the medicine to a new era. Here animation also not working. So now the challenge with the new the personalized medicine, it's to first to decide which one of the patients need to go to which test. So to the right person to the right test, and also to do the right interpretation by combining all this information, and then to finally give the right treatment for the patient. So another question, it's how many personalized medicine are available today? The answer is 140 till I think 6 months ago, and they are in constant increase. And this 140 are FDA approved. So we are, I think, in the point, in the tipping point of a new game, how to develop also drugs. So if we can define a specific and single, like not defined, but classified the cancer or any disease in different classes, we can create a treatment specific for each one. So, and also now is the biopharmaceutical industry, they start playing also the game. So they start understanding that they need to jump in the new era and start developing the drugs and making clinical tests more like specific for the specific mutations or diseases. So if we look to the statistics, actual statistics, 42% of the medicine in development now, 73, they are 42, they are personalized medicine in the development, and 73% of them are the target for a cancer patient. But like any new field, we have some challenges that we need to face to really make personalized medicine a success. The first one is to educate and give awareness for the population, for the patients, and also for the practitioner. They need to learn and educate and be like update their information and to know if there is a new drugs, and also there is the value recognition, also the challenge of to recognize the value of the drugs, and also the infrastructure in the country, like Dr. Dermime said, the countries need to put more effort to create the platforms, for example, for genomic, for diagnostic, to benefit from this personalized medicine. And also the insurance company also need to be part of the game to also approve and take this kind of new drugs or this new technology in account in their system of reimbursement. So it's a personalized medicine. It's a chance to make a revolution for our health system, and but it's required a team effort from different group, government, insurance companies, the patients' education, and the practitioner and the doctors, hospitals. So now I will give a small example of how we can use this genomic and bioinformatics and personalized medicine. So we receive in the clinic a young girl, 30 years old, Arabic, which she was pregnant for the second time, and she delivered already previously one time, para one. She don't have any abnormality in the first trimester. At 30 weeks of gestation, we observe in the ultrasound fetal diffusion, and intestinal dilation with honeycomb sign, and polyderminosis with an amniotic fluid index 28 centimeter. So the family history of this patient, 4 years ago, she got pregnant also, and and she got the same problem when they did the ultrasound. She got the same observation, and this previous pregnancy, she was like till the term, and she gave birth to a child, but this neonate suffer from persistent watery diarrhea, electrolyte it decrease, and poor feeding since the birth. Finally, this baby didn't survive, and we could find a specimen of umbilical cord blood in the hospital, so we could use some this blood for some further analysis. So the clinicians suggest to do whole exome sequencing, and for the child who didn't survive, and for the family, for the father and mother. So we received the samples, the blood samples from the father and the mother and the child, and we entered it to our automatized pipeline, where we create the library and we sequence these samples. We check the quality, of course, and we look for variation, SNP and indel or insertions, and we annotate this variation using a CleanVar or SNP-DB. So the first, the child who died, we found that in his genome, we in his exome, 6 pathogenic variation, and if we compare it with the father and the mother, here I don't know if you can see it clear. So the P, it's the proband, the child, the mother and the father, and we can notice that one of the variation, it's homozygous for the child and heterozygous for the parent, and it's bioinformatically it's classified like high impact, and in CleanVar it's classified by pathogenic. And if you can look to what is the disease classified, it was diarrhea also. So it was a first. So if we look for the term diarrhea in this all the variation we found. Well, we found another variation also. Here it's homozygous also, but this one it's considered like benign, but it's also linked with the diarrhea and some disorder in the intestine. Okay, like we did after to confirm this Sanger sequencing for the first child that is dead and the new child. So we got some samples for the new child, the father and mother and the new child. Okay, and we can confirm that the first child, it's homozygous for this variation where we have AA, and for the father and the mother, it's heterozygous. Here it's the father and here it's the mother, where we have the A and the G, it's heterozygous. And the new child that she's like carrying now, he's exactly the same thing with the previous child. So we found that this parent have a heterozygous mutation in one gene classified pathogenic and linked with this disease causing the diarrhea, severe diarrhea. And by chance, when they make conception of the new the child, they got previously and this time an homozygous for this mutation, and it was like fatal for the first baby. But we suggest no. So in conclusion, that the parental diagnostic, it allowed for the team to intervene and inform the parents earlier about the condition of the fetus. And we can prepare the staff that postnatally it's important to provide immediately salt substitution therapy to the newborn to avoid like the previous child to decide. And for the further conception for this parent, we suggest to them that to get to use the IVF and where they will discard the eggs with homozygous for this mutation and keep only those they are heterozygous for the or homozygous for the normal SNP. So for conclusion, we can say that personalized medicine can shift our emphasis in medicine from reaction to prevention, like I show in this example of the couple, direct the selection of optimal therapy and reduce the trial and the error prescribing wrong medicine or drugs, help to avoid adverse drug reaction by choosing the right combination for the right patient, and also increase the patient adherence to the treatment. If the patient sees that the treatment is effective, so he will continue and not changing in the middle of the course, and improving the quality of the life, revealing additional or alternative use for medicine and drugs candidate, and especially help to control the overall cost of healthcare. And thank you for your listening.
You hear me? Yeah. Thank you, Dr. Belhocine, for this nice overview about personalized medicine, which is definitely is it changing the treatment and also managing the risks for families who have predisposition, genetic, potential for diseases like breast cancer or colon cancer and different other diseases. So if they know their genes and their mutation they have, they can really even prevent diseases from happening. And I know a few cases like that. I know a person, she was working with me and she found out that she had a mutation for BRCA, for example, and she went and removed her breast because her mother died from breast cancer and her grandmother died from breast cancer. And so this way of preventing also diseases, yeah. But also, Mike, I think question to you also that also personalized medicine now is also creating more tests, to be for drugs also. That means that companion tests for each drug now must be performed before giving the drug to any patient to know if that person really need that drug A or B or C, like for breast cancer HER2, for example. They will do the, few immunohistochemistry and they will find out if that lady, for example, has, if she's +3, for example, HER2, that means that she will be treated with Herceptin. If she is not +3, she will not be treated with Herceptin based on the test. So companion tests are now important and must think, yeah. So, okay, let's see the questions. Okay, throughout the history, people used to heal themselves using local herbs, food, linking the person to his environment. Shall this process be part of the future personalized medicine? Thank you, Dr. Belhocine.
It's a good question. In reality, since a long time, for example, me, what I notice in Algeria, many students for their final like PF or doctorate, they go and search for herbal or traditional medicine and take this herb or food and check for what is the active component creating this feeling that it's when they take this herbal they are treated, and try it in a small clinical or in the animals, and if it's working we create a medicine from it. So I think yes, the local herbal food and that we are using in our like traditional in our villages can be a help for personalized medicine to target a new drug, to find a new drug or to find new potential molecules that can help our health system.
The next one, I don't see the next one, but I can go to the other one. Okay, another question said, concerning personalized medicine, this is from Emma, molecular-wise speaking, okay, manipulating cells and drugs won't be harmful to our DNA heritage for the long term, since regulation system is kind of complex and try to resist or escape many times.
Just to separate the ideas here, personalized medicine, it's not manipulating the DNA. This is, if you are talking about CRISPR or gene manipulation, it's a different kind of medicine. So the personalized medicine, it's more about to find the right test to the right patient and to give the right drug. So we are still in the old medicine without manipulating any genes or DNA. So for long term. So if you are talking about modifying our DNA, it's the CRISPR or manipulation gene. I didn't talk about this, but if you want, I can answer. We don't know. It's a new field. It's why it's not approved anywhere to manipulate the DNA directly. It's only in few countries where it's under highly observation, like to manipulate the genes. There is only one guy, he did CRISPR in the futures, I think in China, and he's in jail.
May I add just one thing about manipulating DNA? I mean, there are people with certain mutation when they are born with a certain mutation. So now there is gene therapy now is growing to go and fix that mutation, that error in the DNA. So that field is mushrooming now and getting bigger and bigger, and all pharmaceutical companies are working on that.
Yes.
Okay. Okay. Let's another. I'm looking from the Excel sheet now. Okay. Okay. He asked. Okay. Can you share with us, please, some databases for interpretation of sequencing results?
Interpretation of what?
Sequencing results.
Yes, you have the DB-SNP for the different variation, polymorphic or pathogen. And you have, if you want the clinical part, you have ClinVar It's the most bigger and comprehensive and used by all the pipelines in general now. A CleanVar contain curated and confirmed results about each variation, which disease, and there is even a publication associated if you want to see the details. So I suggest to use CleanVar because it's the more complete.
Okay, another question from Arab Iman said, okay, RNAs emerge as diagnostic prognostic biomarker for cancer, considering their differential expression from a cancer type to another. Is the RNA targeted gene therapy possible in the near future?
You enter in my domain. My PhD was in long coding RNA. So I was one of the first people who work in long coding RNA in leukemia. So yes, if for diagnostic, I can say yes for the moment, and prognostic, not yet. I don't know. Maybe there is, but for diagnostic, I can send after my article that I published in leukemia, where we can use the link RNA to diagnostic which kind of cancer, leukemia, and we found even some leukemia. Reality, the oncogene, it's activated or controlled by link RNA that was expressed abnormally in these cells. So if you need more information about this, I can send the article after by email to this person.
Okay, let's look at the next question. Okay, the next from Hafida. Question said, so we need to know his or her genetic sequence looks like before starting the treatment, and it is different from person to person. And what if we give them the treatment by genetically and don't work? Is it possible?
So here I think there is misunderstanding. We don't give the medicine according to the different genetics. The genetics here, it's to find what is the genesis of the disease, why the disease came. For example, if we are in leukemia, we can try to find what is the problem, what is causing this leukemia, find the genetic basis, that if it's translocation or mutation, and if we know what is the problem, the mutation, we can understand the molecular basis of this disease, how it's the activation or disactivation of this protein, or make the cancer to be developed, the leukemia to start. So if we know the molecular pathway, we can give a drug to regulate this pathway, stop it, or to bring it back to the normal. So it's not according to the genetics in general. It's to find the mutation that drag this cancer, and after we give the medicine to stop this kind of cancer.
Question from Aminat Jani. How can personalized medicine be used to improve the quality of life for patients with autoimmune diseases?
Autoimmune diseases. So how I can answer this? So if we know the basis of this autoimmune diseases, if we do the sequencing genome and we found what is the mutations and what is the molecular basis of this disease, we can produce some drugs, or if it exists, drugs to target this pathway and to block this autoimmune. For example, I give there is a news 2 months ago It was for diabetes type 1. Diabetes type 1, it's considered like autoimmune, like it's the immunity who attack the beta cell and kill them, and we become like diabetes type 1, where the insulin it's not produced. Khalas. So by analyzing the data of the patient, and if we found that he have this autoimmune system, we can give a new drug. It's called anti-CD3, and we can block this autoimmune to enter in action and to stop this disease from killing the beta cell. And we are like stopping the disease, the type 1 diabetes, from appearing from the beginning, from the origin. So we can save millions of people by just stopping the type 1 diabetes from the beginning. And I found this, it's very like really a big news, because as you know, the diabetes, it's not diabetes by themselves, it's dangerous, but it's by all the associated disease around this disease, that cardiovascular, cholesterol, and cholesterol, all this, the other disease that come and added to the diabetes. So if we stop diabetes type 1 from the origin, we will reduce all this cost and all this diseases also from appearing. So yes, this is an example of, for example, for type 1, if we can know that this patient have this ability autoimmune against the beta cell, we can stop it from the beginning before. So, and here we avoid the disease before it starts.
So may I just, maybe the last question, which is related to our, I think, country and continent, you know. I mean, since that this personalized medicine need sequencing, need proteomics really to know what type of overexpressed protein or gene mutation. So this technology is not available in our country or the continent, except I think in South Africa, for example, they have a good sequencing facility, and a few other countries in the South, North Africa, all from Egypt to Morocco, they don't have even next generation sequencing. So how you see this will be achieved in the developing country? And I know that sequencing a kind of cost is going down from billions to now is going to $100 or $200, a few $100 now.
Yes.
So how do you see the future of personalized medicine in our area?
Like, yes, just to correct one information, there is a new platforms appearing in Algeria or Morocco, Tunisia. In Algeria, for example, in Oran, 2 months ago or 1 month ago, they opened a new platform for genomic, where they have Illumina sequencer, and they are working with the hospitals to develop the pipelines. It's not yet in action, but they are doing all the behind work to start soon. And this is a good news. And I think there is some sequencer in Constantine, maybe in Algiers, but they are not in action. But Oran, I know that they're doing something. Rapidly, Dr. Nadia, for the sequencing now we have we have new technology, Nano Oxford, that it costs only $1,000. The sequencer is just small like this. It's $1,000. And you can plug it in your computer, actual computer laptop, and you can sequence less than $100 full with the flow cell, a full genome. And they have also a software. They can analyze the data in the go. When you are sequencing, the analysis it's going and give you the which mutation. And this new technology will really revolutionize, and it give access to this new technology to some country where the infrastructure is not really developed. And I hope that it's in the near future, our country, for example, adapt this new technology and try to bring it
Awesome. Thank
Okay,
you, Dr. Belhocine, Dr. Dermime. We're going to take a 2-minute break, go grab some water, شربوا شوية مية ولا, and we come back and we have 2 excellent presentations by Dr. Sihem Boudina and Professor Nabil AOUFFEN. So we'll see you in 2 minutes.
Thank you.
Don't go anywhere. Bye.
Thank you.
And welcome back. And we can get started. Dr. NAIT Mohamed مع الدكتورة سيهم بودينا. إليك الخط.
Yes, sure. Assalamu alaikum. So thank you very much. So now we are very excited to have with us Dr. Sihem Boudina, who is actually an associate professor in the Department of Nutrition and Integrative Physiology at the College of Health in the University of Utah in the United States of America. She is the chair of the College of Health Councils and also the chair of the MD Ph.D. admission committee at the University of Utah. She has been recently named the chair of the NIH Integrative Myocardial Physiology Pathophysiology A, MPPA study section. Her research focuses mainly on understanding metabolic and cardiovascular diseases and their molecular mechanisms. As scientific records, she authored over than 60 peer-reviewed research articles with more than 10,500 citations as for today. And the title of her presentation is The Molecular Mechanisms Driving Diabetic Cardiomyopathy. So Dr. Boudina, thank you very much to be with us today. You can share your presentation and you have 30 minutes of time. Thank you very much.
Okay. Thank you. Can anyone hear me here? Can you hear me well? I'm not sure if I can just have a sign from anyone that you can hear my voice. Okay. So good morning, everyone. Well, it's still too early for me. So good morning or good afternoon for people who are tuning up from Algeria. First of all, I would like to thank the organizing committee for this kind invitation, and also specifically Dr. Nadia Boutawi. I would say probably I congratulate Dr. Belhocine for an excellent presentation. I think although my topic is probably not going to fit very well with the precision medicine, I would love any insight on how we can use precision medicine for the treatment of non-genetic diseases diseases that are affecting, I would say, a big number of people. And one of those that I would be talking about today is diabetes. And I don't know how can I advance my slides? Because I don't see oh, here we go. Yeah, so just to give you a brief overview of what my lab does. So we are really interested in deciphering the mechanism for, and I would say mechanism, I'm really going to dive into the very detailed molecular mechanisms of diseases. And for a particular reason today, I'm focusing on diabetic cardiomyopathy. But my lab has also worked on adipose tissue biology as well. And I was trained as a bioenergeticist, so I have a lot of background in bioenergetics and biochemistry. So my favorite organelle is mitochondria, FYI. So why diabetes? Diabetes, of course, is a pandemic, I would say. In a very simple word, at least in the United States, it affects more than 34.2 million people, and it really is a big cost for the health cost here in the U.S. It's reaching $88 million to treat this disease or to manage this disease. Unfortunately, there isn't any sign for the disease to be slowing down, but rather there is a very good positive correlation between overweight and obesity and type 2 diabetes. And with the population of the US being mostly overweight, there is a trend toward an increase in the incidence of diabetes. Next slide. So as I prepared my presentation in a very, very short notice, I was hoping to find some statistics on the incidence of diabetes in Algeria. And I would say for the most, I looked at the website of the International Diabetes Federation, and that's the only probably platform that I found. Maybe some people know of another platform, but that's the only platform that I found that gave some estimates for the incidence of diabetes in Africa. And as you can see here, this map is really depicting North Africa and also the Middle East. And one would be happy that Algeria has probably less than 8% of the population affected by diabetes. But I think a lot of it is due to the undiagnosed prediabetes. We know that we have a pandemic of overweight that is actually becoming a problem in Algeria. And with this, of course, comes also an increase in incidence of diabetes. So I think it is still a problem to be concerned about. So I want to, before I go into diving into the diabetic cardiomyopathy, I'm going to just take a break and define what diabetic cardiomyopathy is. It was firstly identified by Shirley Rubler, and this is just after an electron microscopy examination of a heart from a diabetic patient. And what she stated in her concluding remarks is that structural and functional abnormalities are actually observed without really a coronary artery disease or hypertension. So most often when we talk about cardiovascular disease, we think of, stroke, myocardial ischemia, and those are all actually linked to your vessels being clogged. In this case, in the diabetic cardiomyopathy, it's really mostly, I would say, the heart that affects the cardiac muscle as opposed to the vasculature. Next slide. And if we, sorry, I think my slides are just getting messed up a little bit. So if we separated cardiomyopathy word, the cardio, of course, is depicted in the heart, but the myopathy here is really, it's the muscle weakness. Next slide. So my lab worked extensively on trying to, understand how this disease of the muscle occur. And of course, I'm not going to limit this to like one particular mechanism, because there are several mechanisms that we can attribute or several mechanisms that can contribute to diabetic cardiomyopathy, and those are depicted here. But what I'm going to be talking about and what my contribution was for the field is I'm going to talk about the contribution of mitochondrial dysfunction and also the contribution of autophagy in this process. So as you all know, the heart is composed of cardiac cells, but also of a lot of other cells, endothelial cells, fibroblasts, nerve cells. And if you look at an electron microscopy image of the heart, you can clearly see the presence of mitochondria in between these cardiomyocytes. And the human and the mouse heart contains about 23 to 32% of its mass as mitochondria. And this is not without a reason, of course, because as you may know, the heart cannot afford to stop. It's continuously beating. And for that, it really needs a lot of energy. So it's deriving its energy, 95% of its energy from ATP produced in the mitochondria. So my lab initially started really looking at what happens to mitochondria in the diabetic heart. And for that, we did a lot of studies in animal models, mainly in rodents. In this case, I'm showing you some data that we obtained in a diabetic model called leptin deficient model, where this model has a deficiency in leptin, the hormone that can tell you to stop eating. And these animals, as you may see, they are very obese, so they develop obesity. And this is due to the fact that leptin is no longer there to suppress eating. And they become hyperphagic, so they eat a lot. So when we took the heart of those animals and examined mitochondria using biochemical assays, we can clearly see that the mitochondria has, it's impaired in its function. But what most importantly, what we found that despite the fact that it could actually maintain a certain level of oxygen consumption, this oxygen is not necessarily used to make ATP. So that's when we introduced the concept of mitochondrial uncoupling. Next slide. So if you take a heart from those diabetic animals and obese animals, and you compare them to a non-obese and non-diabetic animals, what you can find that the obese animals, their heart is actually spending more oxygen to make less ATP. And that will reflect into having less cardiac work for the oxygen consumed. And this creates, again, a situation of mitochondrial encoupling and also of a cardiac inefficiency. So the heart is still actually working, but it's not efficiently using its oxygen to make ATP. So if you plot, so that's one finding we found. Now, what drives this encoupling? The first thing that we noticed a while back during my postdoc is you would assume that for type 2 diabetes, you are in a situation where your tissue is not really sensing insulin very well. And that's what we call insulin resistance. And if you develop insulin resistance, so your tissue is not really bringing in glucose in response to insulin, because that's what insulin does. It's an anabolic hormone that will allow you to bring in glucose into your cell. So if you are not sensing insulin, then you're not able to bring that glucose. And that reflects here into a reduction in glucose oxidation in the diabetic heart. And because of that, the heart tried to compensate, because the heart cannot, again, afford to be without substrate. So the heart will compensate. And in the instance of type 2 diabetes and obesity, because you have lost this insulin action also in adipose tissue, so insulin has two actions. It actually brings glucose in muscle tissue, but it also suppresses lipolysis in the adipose tissue. So it suppresses the release of free fatty acids. So if you lose the insulin action, then you have two things that happens. In the muscle, you don't bring in glucose, and in the adipose tissue or the fat, you don't suppress lipolysis. And if you don't suppress lipolysis, then you continuously have this flux of free fatty acids in your circulation. And when the heart sees those lipids, it will just take them. The heart does not have a machinery that can regulate the lipid uptake, because it just uses whatever substrate it can see. So consequently, with the decrease in glucose oxidation, you can see a proportional increase in fatty acid oxidation. And that's a hallmark, again, that was confirmed by many, many other groups that we discovered initially in the diabetic heart. Next slide. So as I said, despite the fact that you take those fatty acids and you oxidize them, and you use so much oxygen for oxidizing them, because again, from a biochemistry point of view, fatty acids, as opposed to glucose, are much, much more less efficient substrates compared to glucose. And the reason why is because you consume too much oxygen to produce ATP from fatty acids. And that reflects here in the amount of oxygen you will be producing. So the diabetic mice have much higher myocardial oxygen consumption in the middle panel here, but for less work. And that reflects into a decrease in its cardiac efficiency. Next slide. This was further confirmed by another group in humans, by Dr. Peterson, and she also took women who are obese and diabetic, and she clearly showed that myocardial oxygen consumption as well as myocardial fatty acid oxidation are increased, and they correlate with the area under the curve for glucose, which reflects how much insulin sensitivity they have. So the higher the glucose area under the curve, the more insulin resistant you are. And so she showed also that cardiac efficiency in this subject is reduced as well. Next. Now, my colleague, Dr. Brian Fink, he to confirm that this contribution from fatty acid oxidation is really a problem, What he did, he actually overexpressed a gene called peroxisome proliferated activator receptor alpha. And PPR alpha is a predominant co-regulated for fatty acid oxidation. So he overexpressed PPR alpha in the heart, and he can actually mimic the increase in fatty acid oxidation that occur in diabetes. And that's by itself, in the absence of diabetes, can actually cause cardiomyopathy. Next slide. So that led us to put forward a model to explain, or at least, explain the early changes in substrate switch in diabetes, as well as mitochondrial uncoupling that occur in the type 2 diabetes heart. So we, of course, all what I showed you so far was on type 2 diabetes mouse model. I'm going to switch gear a little bit to tell you a little bit about type 1 diabetes. And as you know, most of you know, type 1 diabetes is really when your beta cells does not work at all and does not produce insulin. I forgot to mention that in the type 2 diabetes, it's the opposite scenario. So you don't sense insulin, but then your pancreas is just trying to compensate by producing more insulin. So we have a hyperinsulinemia in the type 2 diabetes situation, whereas in the type 1 diabetes situation, you have lack of insulin, but then remember that you're going to be injecting insulin from exogenous. So you are still at a state of higher insulin because of exogenous insulin supplementation. Contrary to the type 2 diabetes situation, what we found in a mouse model of type 1 diabetes called Akita mouse, which is just carrying a genetic defect in a key gene that produces insulin. So they don't produce insulin. You can potentially see that contrary to the type 2 diabetes heart, despite the fact that they are still actually consuming a lot of fatty acid, their oxygen consumption does not actually change. And therefore, their cardiac efficiency also doesn't change. We still don't know exactly why is that, but most probably we think that you still have an increase in free fatty acids. However, you probably don't have a higher hyperinsulinemia that we see in the type 2 diabetes. And that could be maybe driving some of the defect in mitochondrial energetics. Next slide. So this is just a comparison side by side for the type 2 diabetes and type 1 diabetes. And what we saw is that both of them actually have an increase in reactive oxygen species. So when the mitochondria relies too much on fatty acid oxidation, and it's bringing too much electrons into the electron transport chain, the electron transport chain feels bombarded by these electrons. So some of them will skip the electron transfer and they will go into making reactive oxygen species. And to really try to mediate this oxygen consumption that doesn't actually fully go into ATP production, but rather to making reactive oxygen species, the heart can actually turn on proteins that can mitigate the membrane potential. And those proteins are called uncoupling proteins. One of them is UCP3, which is depicted that not to be changed in the type 2 diabetes, but it's upregulated in type 1 diabetes. So it is possible that, sorry, go back one more slide. If we can go back to the previous slide. So it is possible that the type 1 and the type 2 diabetic heart is dealing with different oxidative stress load, and it's responding differentially to this oxidative stress load. Next slide. The other thing, of course, that we try to compare type 1 to type 2 diabetes is the insulin sensitivity. So you may imagine that if you do no longer, respond to insulin, your downstream signaling of insulin receptors will be downregulated. This actually surprised us when we look at, for example, AKT, which is a downstream protein downstream of insulin receptor signaling. So when insulin binds to its receptor, it activates a cascade of signaling pathways, and one of them is this AKT pathway. So if you take a wild-type heart or control heart, you give it insulin, you see an increase in the phosphorylation of AKT. This does not occur in a type 2 diabetes heart, okay? If you take, in contrary, if you take a type 1 diabetic heart, okay, you give it insulin, like this here, Akita mice, you give it insulin, it's still actually responding correctly to insulin. So that could be one big difference between the two models here, is the fact that, type 1 diabetes mice still actually respond appropriately to insulin, whereas the type 2 diabetes don't. Next slide. I told you before that, this insulin sensitivity and downstream signaling may be important for mitochondrial respiration and mitochondrial energetics. So to further confirm that, what we did is we used genetic, we used the Cre recombinase system to generate a conditional knockout that is lacking the insulin receptors specifically in the heart. So we took mice that were where the insulin receptors was flux by 2 LOXP sites, and we crossed it with an alpha MHC Cre. With an alpha MHC is a myosin heavy chain that is exclusively expressed in the heart, and we generated mice that do not have the insulin receptors specifically in their cardiac cells. And Dr. Belki from Dr. Abel lab has shown that, yes, these mice, they don't have the insulin receptors in the heart, and they also don't respond to a bolus of insulin injection. Next slide. When we took their mitochondria from the heart and really look at their mitochondrial oxidative capacity and ATP generation, we can clearly see that if you just lack the insulin receptors by itself in the cardiac cells, that's sufficient to cause a decline in mitochondrial respiration and ATP production. And again, those mice don't have diabetes, okay? I just want to emphasize they have no differences in their free fatty acids or anything else. They just lack the insulin receptor in the heart. And our objective was really to test whether just removing the insulin receptors from the heart is sufficient to cause a mitochondrial dysfunction. So that was indeed the case. Next slide. So we also showed that just by manipulating the insulin receptors from the heart, you almost create a situation like the type 2 diabetes situation. So these mice rely heavily on fatty acids, and they also have an increase in oxygen consumption, and they also have a reduction in cardiac efficiency, like the type 2 diabetic heart. So at the end, we could potentially conclude that with this diabetic state, and especially the type 2 diabetic state, there is, first of all, there is a shift in substrate oxidation. So the heart sensing that glucose can no longer enter the cells normally, it has to bring in something else. And that something else is really dependent on what you have in the circulation. So in the type 2 diabetes, because there's abundance of free fatty acids circulating because of the lost suppression of lipolysis, then the heart will use those fatty acids. And as a price for just relying on fatty acid oxidation, you have a reduction in your cardiac efficiency as a result of mitochondrial uncoupling. So I'm going to switch gear. Again, I told you before that I'm going to be talking about mitochondrial dysfunction as a mechanism for cardiomyopathy. And now I'm going to really switch gear to the autophagy pathway. And there is a link between the two because, first of all, let me just drive you through the autophagic process. So autophagy, auto is a self and phagy is eating. So it's coming from the Greek word autophagy that means eating itself. So all the cells in our body, all of them, have this process. It's a very, very conserved process. And for cells that regenerate, tulta, it's like, for example, blood cells or some cells that proliferate, cancer cells in this case, they don't really need this process very well, a lot, because they can generate their cells again. In cells that are not capable of proliferating or regenerating, including most of our cells, like neuronal cells, cardiac cells, but also some other cells, which we call postmitotic, which means once they form, they cannot actually proliferate. This process is very important for them because it helps them clean all of the garbage that occur in the cell. When I say garbage, I mean, for example, misfolded proteins, I mean, oxidized organelles, and so on and so forth. So what this process involves is it involves a membrane structure that we believe is originating from the endoplasmic reticulum, and it brings a cascade of signaling pathway that starts with class 3 PI3 kinase, and then it involves a lot of autophagy genes, as you can see here, autophagy 5, 12, 16L1. And there is a microtubule protein called LC3. That microtubule protein is actually the key for making this process occurring. So with the help of an ubiquitin ligase called autophagy 3, this protein LC3-1 is lipidated, which means there is a lipid moiety that is added to it to become an LC3-2 protein. And that LC3-2 protein really gonna tag all of the autophagosomes and make the signal for this membrane structure to actually start curving and start really englobing all of the garbage that needs to be degraded. So you form what we call the autophagosome. So it actually surrounds the garbage and it closes. And then this closed structure gets in proximity with the lysosome. And the lysosomes, as you can know, it's a structures. There are structures in the cells that contains ATPases. So they are just here to chew all of the unwanted proteins. So this process is, of course, is important for not just cardiac cells. It's important for anything. It's a very, very fundamental process. But in the heart, it has been shown that if you really don't make these proteins, for example, ATG3 or ATG5, the heart really progressed into a heart failure, and the mice died. So next slide. So why am I talking about autophagy? How can I link this autophagy to my previous portion of the talk? So I told you that autophagy is a process that is really, really important in the cell. For example, if you starve, okay, you starve, you don't have any food, your cells will sense this situation, and the first thing that they want to do is they want to bring in ATP. So how do they bring ATP? They will have to go back into your cell and look at for things that they can actually degrade to make ATP or substrates. And this sensing of starvation signal is actually done by the insulin receptor pathway. So when you starve, the insulin is not brought to the cell. There is no engagement of the insulin receptor signaling pathway, and there is no activation of a protein called mTOR, or mammalian target rapamycin. And if you don't activate the mammalian target rapamycin, which is a suppressor of autophagy, then autophagy is upregulated. And this is, again, a way for your cell to protect themselves from dying. So if you increase your autophagy, you can degrade components of the protein, and you can recycle your amino acids for usage to make ATP. Next slide. So this process of autophagy, of course, is very important. So for example, if you mess up with insulin receptor substrate, which is the first protein that is actually regulated by the insulin receptor pathway. So if you do that, this is embryonic lethal. Okay, the mice don't make it. Shortly after birth, they die, and they die with a dilated cardiomyopathy. Next. So again, the conundrum here that I want to say is, you would think that if insulin receptor pathway is really a suppressor of autophagy, then if you are in a situation where you have insulin resistance or diabetes, then your autophagy should be, what do you think? I would think if you don't have a suppressive signal, then your autophagy should be upregulated. However, next slide. When we did look at autophagy in the same model of type 2 diabetes, OB mice, next slide, we can clearly see that autophagy is really, contrary to our hypothesis, autophagy is actually decreased rather than increased when insulin action is not sensed. And that was really something that we were really triggered by, because the whole point was to, try to demonstrate that the reduction in autophagy is really contributing to the lack of cleaning the garbage, and that actually contribute to cardiomyopathy. But what we found, contrary to what we found, is we found that the insulin receptor signaling is actually upregulated in type 2 diabetes mice rather than decrease. Okay, so if you look at basal AKT phosphorylation, basal, phosphorylation of mTOR, they're all upregulated. Okay, next slide. So the first question that we had is, maybe because there is an increase in AKT phosphorylation, there is an increase in mTOR phosphorylation. This is really what is driving the suppression of autophagy. So next slide. So we wanted to target mTOR. And what we did here is we treated mice with the mTOR inhibitor rapamycin. And when we did that, we would clearly see that the suppression, inhibiting mTOR was actually really key for the reduction in autophagy in these hearts. So mTOR plays a role in suppression autophagy in the type 2 diabetic heart. Next slide. So how that can occur, because of course, you are in a situation where you have insulin resistance. So why are these hearts still responding to insulin? So one idea that we had is we think that maybe, because of this compensation in insulin that I told you about from the beta cells producing extra amount of insulin, we went and we look at circulating insulin levels in this mice, and you can clearly see that their insulin levels were to the roof. So they are very, very high. So they have a very frank hyperinsulinemia. And insulin receptors structure is very, very similar to. Sorry, I'm just losing my thoughts here. Just a minute. So because of this hyperinsulinemia, in the cell, you have two receptors. You have the insulin receptors, but also you have the IGF receptor that are very, very similar in structure with the insulin receptors. And it has always been taught that high level of insulin will activate the insulin receptors. High level of IGF will activate the IGF receptor. However, what we are demonstrating here is we are demonstrating that if you have a very high level of insulin in your bloodstream, you can also activate the IGF receptor in the heart. And this is shown here by activation of IGF receptor. This, when we did this study, there was another study that was published at the same time that also showed that in patients with type 2 diabetes, they also have this hyperinsulinemia, and they also activate downstream signaling of AKT. Next slide. So we hypothesized then that it's maybe the activation of the IGF receptor by systemic hyperinsulinemia that is responsible for the suppression of autophagy. So yes, you don't respond to insulin, but you're able to respond to IGF, to insulin by IGF receptors, and that's actually is sufficient to suppress your autophagy. Next slide. So what we did is we took this obese mice, and we made a genetic manipulation where we actually deleted one allele of the IGF receptor. So we made them heterozygous for IGF receptor in the heart, okay? And when we did that, we clearly can see that we can suppress AKT signaling. So meaning that the IGF receptor was responsible for this increase in AKT signaling in these mice. Next slide. However, when we look at autophagy, we really didn't see a full rescue of autophagy when we removed the IGF receptor. Next slide. So we concluded that maybe this suppression of autophagy then is not consequent to the increase in IGF receptor signaling. So what we did next is we look at ERK signaling, because ERK was upregulated as well in this heart. And when we suppressed ERK signaling, we can clearly see that really by suppressing ERK, there was a suppression, a rescue of autophagy. So in conclusion, what we concluded that is hyperinsulinemia is sufficient to increase AKT signaling, and it's probably responsible for the cardiac growth that we see with diabetes. However, it's not contributing to the suppression of mTOR and autophagy. And the suppression of mTOR and autophagy is probably through ERK, because ERK is known to directly block mTOR, and activation of probably some oxidative stress or inflammatory signaling is responsible for the upregulation of ERK in the diabetic hearts. So I don't think I will have time to go. I don't know how am I doing in time?
Yeah, I think we don't have time for that, so we can take questions.
Let's skip one. Let's skip this one, and I think if you can skip the slides, I may have just a conclusion slide at the end. Yeah. So the take-home message here is that although systemic hyperinsulinemia activate IGF receptors in the heart and participate in the growth of the heart through AKT, it's not involved or does not participate in the impairment of autophagy in this diabetic heart. And with that, I would like to, next slide, just thank the people who did the job. Next slide. And I will take any questions. Sorry for bombarding you with a lot of information here.
No, thank you very much, Professor Boudina. It was really exciting and helpful. So far, as you know, there's a lot of PhD students that are with us today. So I'm sure that they got a lot of information from your work. Thank you very much. Yeah, so I like also to ask people in the audience if they want to ask questions, and they just have to write them in the chat. By the way, we have some questions that pop up during the presentation, so I can start. So are any personalized therapies approved for the human diabetic cardiomyopathy?
That's an excellent question. I think that was one question that I asked Dr. Belhocine, but I think they didn't have time to bring it to him. There is a component of genetics, of course, for this diabetes incidence. Okay, so type 2 diabetes, I would say, is more of a lifestyle disease. So it's linked to, again, the incidence of obesity and the way you eat, the way you move. Type 1, however, of course, I would say there is a big component of genetic in it. So I think there are now identified SNPs and identified genes that are actually increase your diabetes incidence. And those genes, I think, could be maybe a subject of trying to develop personalized medicine for these patients. For example, I'll just give you maybe our group has identified a genetic mutation in the adiponectin receptor. And as you know, adiponectin is an adipokine that is secreted by adipose tissue that maintains insulin sensitivity. So people who carry this adiponectin mutation, they are prone for developing diabetes-related nephropathy. So it is possible that if we can maybe do genetic screening, whole genome sequencing to really identify these mutations, study their function, and then hopefully maybe develop either genetic therapies or understand the pathways downstream of those and do targeted therapies for these pathways. So it is possible.
Thank you very much. One question from the audience, from Dr. Boudjelal, actually, is as ATP has a role in both, as you said, diabetes 1 and 2, and also is an important factor for cancer cell to grow fast. How do cancer patients with diabetes 1 or 2 looks compared to cancer patients who do not have diabetes?
Well, first of all, I should say I am not a physician, so I can't really speak too much about the clinical manifestation of the disease in the cancer population. My simplistic thinking is that, if you think about the insulin receptor, so one thing that I would say that reflects what I showed you so far, there is a very, very positive correlation between hyperinsulinemia and the incidence of some cancers. And we think that maybe this is really through this IGF receptor. IGF receptors are pro-growth, so they promote growth. And cancer cells loves actually the IGF receptors or the insulin receptor. So they love pro-growth signaling. So that's one thing I would say, that if you have maybe hyperinsulinemia as a cancer patient, you might actually have more aggressive tumors because of this pro-growth signaling that is upregulated.
Okay, great. Yeah, we have also a question from the audience, Emma Mardin. She said, since diabetes is related also to cancer, do you think regulating this would refrain the neocancer frequency if working on those receptors? So, as you said, IGF receptor, since the most of them are also related to sugar metabolism directly and indirectly.
Yeah, I think we did answer this question somehow, because again, if I think if we can find ways to minimize the overproduction of insulin from the beta cells in an instance of type 2 diabetes, that's probably going to be a good way to go. Okay, because again, it's the hyperinsulinemia, I think, that is driving most of the signaling, because it's trying to compensate for something. And in the instance of the heart, it's also trying to compensate for the lack of insulin signaling by upregulating this AGF receptor. And what it does, we actually clearly showed that if you upregulate IGF receptors, you increase AKT and you increase cardiac growth. So it's possible that in the cancer cells, this pro-growth signaling is going to make the cancer cells, grow even more. So so that's my simplistic view of this. But again, I'm not a clinician. I can't really speak of, how the cancer patients have, will do with or without diabetes.
Yes. Also, one other question that came in the audience when you were talking about autophagy. Maybe you already got that question a lot of times. So, as you talked about autophagy, what are the benefits of the intermittent fasting, like for example during Ramadan, on diabetic patients?
Well, that's an excellent question. So I did not mention that, one of the Nobel Prize in Physiology was attributed to a Japanese scientist who discovered autophagy. And the way he discovered autophagy was through actually, starvation. So, so with the intermittent fasting now, what we're finding, I don't study intermittent fasting, but I have a colleague who study time-restricted feeding, which is kind of the same thing as intermittent fasting. So it's believed that if you fast for at least a period of 16 hours, you turn on autophagy, okay? And because you're doing it not all the time, because you don't want to activate autophagy all the time, okay? You don't want to activate it all the time. If you super activate autophagy, remember, it's eating your cells. So you could potentially have cells that will die. So this intermittent increase in autophagy and decrease is almost like a signal, a protective signal. Okay, so it will allow you to get rid of the unwanted protein or garbage, then go to a steady state. And then when you accumulate more garbage, you go on and you clean it and you go on to a steady state. So that's what we think intermittent fasting is doing. It's boosting this autophagy in a very, cyclic way. And that's, in my mind, is very beneficial. But of course, if we talk about Ramadan, and if you're doing Ramadan for 16 hours, but then you are eating a lot of sugary food, I am not 100% sure that your autophagy would be optimal.
Awesome. Thank you so much, Dr. Boudina. What an honor.
Thank you.
Yes, thank you very much again, Dr. Boudina. So I think we will move to the last but not least speaker. So he's there actually. Great. So I have now the pleasure and also the honor to present our speaker from Algeria, Dr. Nabil AOUFFEN, who is actually a full professor in medicine. He is chair of the Anesthesia and Intensive Care Department at Ahmed bin Bella University at University of Oran. He is the former director of the Thematic Research Agency in Health and Life Sciences, more easily known as ATRSS. He sits on the College of Experts of the National Economic, Social and Environmental Council of Algeria. He is a founding member and director of the Algerian Journal of Health Sciences, AJHS, a journal where I also publish some of my research paper, by the way. He is also a founding member of the Society of Anesthesia, Intensive Care and Emergencies. He is also a consulting expert for the ATRSSV and the Ministry of the Higher Education and Scientific Research of Algeria. So thank you very much for being with us today and speaking more particularly about the personalized medicine in Algeria, the interests, the gaps, and the needs. Question came during the first and the second talk about what is actually happening in Algeria. And I am sure that a lot of question and discussion will come after your presentation. So please share your screen with us and you have 30 minutes of time.
بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله تعالى وبركاته. شكرا جزيلا للمنظمين لهذا الشرح العلمي. بادئين ببدء أود أن أقول بأن هذه النظرة الاستشرافية هي نظرة un groupe de réflexion qui constitue de 3 personnes, en l'occurrence Madame Touel Bokofa, Monsieur Ben Saada et moi-même, qui constitue pratiquement le groupe focal Algérie de réflexion sur la médecine personnalisée en Algérie. Alors, qu'en est-il réellement sur le plan pratique en Algérie en matière de médecine de précision? Alors, l'Algérie a pris conscience de ses priorités et le gouvernement a établi pratiquement 3 priorités pour faire avancer, entre autres, la santé du citoyen algérien. Et pour cela, donc, le gouvernement a mis en phase 3 priorités, la sécurité énergétique, la sécurité alimentaire et la santé du citoyen. Pour rappeler un petit peu, dans notre cursus de formation, les médecins doivent avoir 7 compétences. 7 compétences qui sont reconnues par l'OMS et CADMETS. Il doit être expert médical, il doit être communicateur, il doit être aussi collaborateur. Et on va voir par la suite pourquoi je vous dis ça. Il doit être leader dans son domaine, il doit être aussi promoteur de la santé, il doit être érudit et professionnel. Tout cela va avoir une incidence et une implication et une corrélation avec la médecine personnalisée. La médecine personnalisée qui a été appelée dernièrement médecine 6P, 6P parce qu'elle est personnalisée, elle est de précision, elle est préventive, elle est prédictive, elle est pluriprofessionnelle et un parcours multiple, multi-parcours. Donc, vous voyez que c'est extrêmement important la formation par rapport à la mise en place de cette belle opportunité pour faire avancer la médecine personnalisée en Algérie. Alors, réellement, qu'est-ce qui se passe en Afrique? Pour aller faire après un zoom en Algérie, il y a un déséquilibre dramatique dans les données génomiques mondiales. Si je prends l'Afrique, vers 2050, on sera à peu près quelques chiffres à 2,5 milliards de population. Donc, plus de 25% de la population mondiale qui va être représentée par la population africaine. Mais réellement, sur le plan génétique ou génomique, simplement 2% des données génétiques mondiales proviennent d'Africains. Ce manque de diversité aura certainement un impact sur la santé et sur les efforts conscients des inégalités qui vont exister entre l'Afrique et le reste du monde et qui vont s'aggraver au fil du temps et ils vont contribuer certainement à un écart entre ce qui se passe dans le monde et ce qui se passera en Afrique en matière d'innovation, nonobstant de ce qui va se passer en Algérie. Donc, en sous-estimant cette diversité génétique humaine, le progrès scientifique ou le progrès technologique ou l'innovation auront des conséquences néfastes qui vont freiner toutes les avancées de cette médecine personnalisée. Afin de remédier à ce déséquilibre, des données génétiques ou génomiques, l'Algérie souhaite entreprendre des efforts. Et à ce titre-là que je vais vous parler en qualité de point focal Algérie pour le développement de la médecine personnalisée en Algérie. Alors, pour préciser le propos, l'Algérie, elle est en transition, 3 transitions essentielles. Transition démographique, premièrement, une transition épidémiologique et la troisième, une transition socio-économique. Démographique parce qu'il y a un allongement de l'espérance de vie, épidémiologique avec l'accroissement des maladies non transmissibles et surtout la cancérologie, le diabète, l'hypertension. Donc, nous allons cumuler les problèmes des pays développés et les problèmes médicaux des pays qui sont en voie de développement. Et pour cela, la troisième conséquence, c'est le développement socio-économique. Le développement socio-économique va aggraver les facteurs de risque et on va le voir par la suite. L'Algérie, je vais finir le propos de ce qui se passe en Algérie. L'Algérie, c'est trois régions. C'est plus de 2 millions de kilomètres carrés. Il y a la région nord, avec ses spécificités médicales ou épidémiologiques, les hauts plateaux aussi, et bien entendu la troisième région, c'est le sud. Donc pratiquement, on aura à faire des approches particulières en matière de ces 3 régions, en matière de soins et de qualité de soins pour la médecine personnalisée. Comment est perçue par nous et par les décideurs les potentialités de la mise en place de cette médecine personnalisée. Je vais parler en qualité sous l'égide de notre département ministériel qui est l'enseignement supérieur et la recherche scientifique. Il y a une nécessité de faire une collecte et une analyse des données génétiques pour améliorer la découverte scientifique et ainsi, pour conséquence, l'amélioration de la santé et tirer parti de ces innovations actuelles et futures en matière de soins en qualité de santé. La médecine personnalisée mobilisera le triptyque, les chercheurs, les praticiens que nous sommes et les acteurs industriels. Donc, c'est le développement de la recherche et de la formation dans le domaine de la génomique pour l'amélioration de la santé humaine va s'articuler autour de 3 axes principaux. C'est la mise à niveau et ou le développement des centres de recherche, tels que les plateformes NGS, afin de permettre la conduite des projets de recherche dans le domaine génomique, le renforcement des compétences humaines algériennes, bien entendu en soutenant la formation, les stages de perfectionnement dans le domaine de la génomique, l'analyse et l'interprétation des données, en s'appuyant sur la communauté nationale qui est établie à l'étranger. Bien entendu, aller sur des collaborations dans des projets à l'international, particulièrement en oncologie et d'autres. Quelques considérations. La médecine personnalisée a un potentiel énorme, comme vient d'être cité, pour améliorer le diagnostic, et pas uniquement le diagnostic, le traitement et surtout la prévention. Cette médecine personnalisée aura une opportunité pour les citoyens, et pas uniquement pour les citoyens, pour les patients et les chercheurs, et donc nécessite certainement un engagement palpable des décideurs et des politiques pour définir des approches communes, une normalisation de ces approches, de définir les priorités en matière de santé du citoyen et de la recherche-développement. Bien entendu, la mise en œuvre de cette médecine personnalisée en Algérie dépendra de plusieurs facteurs, à savoir une évaluation de la situation globale de notre système de santé, du système national de recherche et d'innovation, une nouvelle approche dans l'élaboration de cette politique, de définir les facteurs sociaux et les pratiques culturelles par rapport à l'approche sociétale. L'objectif, bien entendu, de cette médecine personnalisée, c'est de couvrir les thèmes généraux et les éléments structurels qui vont contribuer à façonner le paysage de cette médecine personnalisée en Algérie. Enfin, en intermédiaire, de discuter quels moyens à mettre en place pour faire avancer le domaine sur les bases d'une approche, d'un diagnostic précis sur la base des insuffisances et des besoins qui doivent être identifiés par les chercheurs et les praticiens. Enfin, in fine, déclencher au niveau national une action, à concevoir une stratégie gouvernementale. Mais cette stratégie gouvernementale aura à prendre en charge les visions intersectorielles, c'est-à-dire le ministère de la Santé, le ministère du Travail, le ministère de l'Enseignement supérieur et de la Recherche scientifique, le ministère de l'Économie, des startups et des micro-entreprises, tous qui vont conjuguer leurs efforts pour faire avancer le développement et la mise en œuvre de cette médecine personnalisée. Alors, entre autres, il y a une réflexion pour organiser un workshop pour la mise en place du réseau thématique de la médecine personnalisée en Algérie. Pourquoi? Parce qu'il s'agit aujourd'hui, chacun dans son petit coin travaille tout seul, les chercheurs tout seuls, les praticiens hospitaliers tout seuls, et dans des écoles dédiées pour la biotechnologie. Ce workshop aura à unifier, grâce à la mise en place d'un comité d'experts qui va identifier les besoins et de définir un petit peu la stratégie et les défis à relever, ainsi que les opportunités à la mise en place d'une collaboration à l'échelle nationale, voire par la suite à l'échelle africaine ou maghrébine, avec la région MENA, avec l'Europe ou à l'international. C'est les objectifs de ce workshop ou de cet atelier auront à examiner dans le détail, avec précision, l'état de l'avancée de cette médecine personnalisée en Algérie. Il va identifier certainement et prioriser les domaines d'intérêt commun identifier nos insuffisances ou nos forces ou nos besoins, nos faiblesses et éventuellement nos menaces pour développer un petit peu les domaines d'intérêt à l'échelle nationale dans le cadre d'une mutualisation et pour construire un consortium national en matière de gouvernance, de financement, d'établissement de partenariats, ainsi que les programmes de formation à l'échelle nationale. Alors, les résultats escomptés de ce réseau, bien entendu, c'est de proposer à notre département de tutelle un plan d'action pour une collaboration éventuellement nationale avec l'Europe, avec l'Afrique, avec d'autres pays éventuellement tels que le MENA ou à l'international avec les États-Unis. L'atelier devrait également servir de plateforme. Il s'agira pour aider les participants à mieux comprendre les avantages potentiels de ce développement en matière de gestion des ressources humaines, c'est-à-dire les compétences en Algérie, en fonction de notre région et des facteurs clés qui peuvent faciliter le développement de cette médecine personnalisée. Enfin, in fine, sensibiliser aux possibilités de collaboration transfrontalière et de stimuler les activités de mise en réseau, qui est importante à notre avis, et c'est la clé de la réussite en matière de médecine personnalisée. L'état des lieux de l'existant en matière de médecine personnalisée en Algérie. Il y a 5 points sur lesquels on peut s'articuler. Nous avons déjà des structures, nous avons des équipements, nous avons des compétences, nous avons eu des financements. Mais est-ce que déjà nous avons notre écosystème qui a été mis en place? Où sont ces insuffisances? Pour répondre, bien entendu, à 2 problématiques qui sont importantes à notre avis, c'est les attentes des praticiens et bien entendu les attentes des patients. Parce qu'il s'agit d'aller vers une qualité de soins pour le patient. Alors, l'existant en matière de recherche, comment cette recherche est organisée? Alors, les recherches en matière de médecine personnalisée, elle a traité divers domaines, la génétique, les études moléculaires et cliniques, l'immunologie, la biologie, la NAPAT, l'épidémio, la bioinformatique et l'intelligence artificielle. Comment elle est organisée? Les unités de recherche sont organisées et sont rattachées le plus souvent à des universités, voire à des centres de recherche indépendants ou à des écoles de biotechnologie, à des agences nationales pour le développement de la médecine ou des sciences de la santé, pour le développement de la recherche en sciences de la santé. Il y a qui sont sous tutelle du ministère de la Santé publique, tel que l'Institut Pasteur, et bien entendu, il y a de la recherche du haut niveau et des équipements qualifiés tels que le CRBT qui sont domiciliés au niveau de Constantine, le Centre de recherche de biotechnologie, et les laboratoires Temeus qui sont des laboratoires labellisés et qui représentent à peu près 9 unités qui ont été labellisées dernièrement par la Direction générale de la recherche scientifique et du développement technologique. Quel est le point de situation en matière de ressources humaines? Si je prends 3 axes, la recherche en cancérologie, la pharmacogénomique et les études populationnelles, vous voyez les chiffres. 230 personnes expertes qui travaillent en oncologie, 21 thèmes de recherche. Pour la pharmacogénomique, 112 experts avec 9 thèmes de recherche. Les études de population, 65 experts, 4 sujets de recherche. Alors, la quasi-totalité des praticiens et des chercheurs sont favorables à l'extension des pratiques en matière de gestion de la performance et de la qualité pour développer des programmes d'études en bioinformatique et en intelligence artificielle qui sera orientée vers la médecine personnalisée. Les plateformes. 4 types de plateformes. NGS, spectrométrie de masse, équipement de biologie moléculaire, serveur et calcul de haute performance. Nous avons pratiquement, pour la génomique, NGS, la protéomique et pharmacogénomique, bioengineering, bioinformatique, la recherche moléculaire pour les antibodies, cell and tissue culture et flow cytométrie, bien entendu, qui est une plateforme qui se trouve au niveau de la CRBT. Je n'ai cité que les centres référents ou les universités référentes. Il se pourrait qu'il y ait des oublis, mais je ne pouvais pas tout mettre sur cette plaque. Notre écosystème national en matière de médecine de précision. Le système de santé est caractérisé par une forte densité d'infrastructures sanitaires, que ce soit au nord, au niveau des hauts plateaux, ou au niveau du sud. Mais les plateformes universitaires hospitalières sont là. Et elles sont appuyées par la Direction générale de la recherche scientifique et développement technologique. Il y a un double régime de financement, le financement par le Fonds national de la recherche et d'autres financements directement par des dotations directes pour le soutien du gouvernement pour le développement de projets de financement de plateformes génomiques et de préautonomiques. Mais il y a une nécessité aujourd'hui d'aller vers des plateformes nationales pour défendre les avantages de la recherche dans le domaine de la médecine personnalisée, et je vais vous en parler. Quelles sont les attentes des praticiens et des patients? Il y a lieu de se concentrer aujourd'hui sur l'utilisation préventive. Je dis bien l'utilisation préventive de la médecine personnalisée et non pas simplement sur les aspects curatifs. Permettre, bien entendu, voire améliorer l'accès de cette médecine personnalisée en matière de soins de santé. Si je dois faire un résumé de cet état des lieux de l'existant en matière de médecine personnalisée, tous les experts, sans exception aucune, estiment que tous les ingrédients sont déjà là, qui sont nécessaires au développement de la médecine personnalisée qui existe au niveau de l'Algérie. La technologie, les compétences, quelques formations et les plateformes. Toutefois, il y a un manque en matière de, où il y a une incompréhension au niveau des décideurs qui fait défaut, avec une vision ou une approche globale qui devrait être nécessaire pour mettre en relation les différents secteurs afin de faire progresser cette médecine personnalisée. Les efforts nationaux sont perçus comme essentiels pour que la médecine personnalisée soit enfin proposée aux patients. Ce sentiment partagé est que la médecine personnalisée développée en tant que discipline, comme le montre le personnel qualifié et tous les travaux de recherche publiés dans des revues indexées à l'international, qui sont au nombre de 1000, mais que les résultats doivent maintenant être mis en pratique en matière de recherche translationnelle. Les thèmes d'intérêt suivants: le cancer, y compris la génétique des tumeurs, les maladies immunitaires, les maladies neurodégénératives, les maladies métaboliques et cardiovasculaires, pour ne citer que cela. La question des tests. Aujourd'hui, les tests apparaissent comme une préoccupation commune de tous les laboratoires de recherche et pour les praticiens, car leur coût est beaucoup plus élevé au niveau national en raison des coûts des réactifs, voire de leur disponibilité. La maintenance aussi des équipements et l'externalisation, voire l'analyse, sont également à prendre en considération dans les limites pour le développement de cette médecine personnalisée. Les activités aujourd'hui à privilégier par toutes les parties prenantes impliquées dans l'écosystème de la médecine de précision au niveau national, par exemple les universités, les ministères, les instituts de recherche, etc. Si je dois parler des équipements et des infrastructures, il faut doter ces infrastructures nationales, hôpitaux, structures hospitalo-universitaires, centres de recherche nationaux, laboratoires, d'équipements de qualité. Et il va falloir aussi faire une cartographie des équipements disponibles et les lieux où ils se trouvent, voir comment essayer de mutualiser pour ne pas dupliquer les mêmes équipements dans la même région et mettre en place des biobanques rattachées à des structures hospitalières ou de recherche. Un projet qui est en cours, de développement pour la mise en place de 3 biobanques en Algérie. Il s'agit aujourd'hui d'avoir une vision transversale et de la mutualisation parce qu'il s'agit de faire une économie en matière de dépenses d'argent pour les équipements qui sont trop chers. Alors, sur le plan formation, il y a lieu d'intégrer l'intelligence artificielle, la bioinformatique et la génomique dans les programmes d'études existants en matière de santé et dans les sciences de la vie. Il faut veiller à ce que les praticiens bénéficient de formations continues, c'est des enseignements post-universitaires. Et dans les programmes universitaires, ouvrir des masters, des doctorats en bioinformatique et de l'intelligence artificielle appliquée à la santé, ainsi que l'analyse de données massives. Sur le plan recherche, il y a lieu aussi de favoriser les synergies entre les réseaux nationaux de recherche pour accélérer le transfert de connaissances. Il faut soutenir la mise en pratique des connaissances en matière de la gestion de la performance et surtout mutualiser les efforts financiers pour développer les projets de recherche collaborative en mettant en place des sujets d'intérêt commun dans une vision collégiale et de partage à l'échelle nationale. Sur le plan politique, il va falloir qu'il y ait un travail de fond à faire pour sensibiliser les décideurs aux avantages de la médecine personnalisée malgré son coût élevé pour l'économie de la santé. Enfin, intégrer tous les acteurs, chercheurs, experts dans les enjeux éthiques, juridiques et sociaux, les économistes de la santé, les patients, l'industrie, à ne pas oublier, et les décideurs politiques, les agences de financement dans leur diversité. D'ailleurs, il y a eu 2 expériences, l'une qui a été réalisée, celle de Pfizer, je n'ai aucun conflit avec ces firmes pharmaceutiques. Pfizer a pris en charge la formation en biotechnologie de la faculté de pharmacie d'Alger et l'école de biotechnologie de Constantine, pour aller sur 3 phases pour essayer de faire des immersions pour la médecine personnalisée. Roche est en train de discuter éventuellement une vision pour mettre en place cette nouvelle dynamique de formation et de politique dans la réalisation de la médecine personnalisée en Algérie. Toutes ces initiatives doivent mettre l'accent sur, avec l'aide des sociétés savantes en médecine ainsi que les associations des patients. Nous avons des forces, nous avons des opportunités, nous avons aussi des faiblesses et nous avons aussi des menaces. Il va falloir y aller pas à pas sur 3 pistes d'action en matière de médecine de précision. Il va falloir qu'on défende nos intérêts. Il va falloir prendre en charge des enjeux éthiques, régler les problèmes juridiques et les problèmes sociétaux. Et il va falloir aussi qu'on prenne en charge la logistique et définir toutes les infrastructures pour qu'on aille vers des équipements adéquats, disponibles au niveau national, tels que réactifs, biobanques, cohortes et la centrale centralisée, la centrale des données et la protection de nos données. La collaboration. Les thèmes de la collaboration doivent être définis. Ils doivent être axés essentiellement sur les maladies. Je dis bien essentiellement et pas exclusivement sur les maladies non transmissibles, parce qu'il s'agit d'aller sur un traitement pour le traitement et surtout pour la prévention. Nous avons le cancer avec sa prévalence ou son incidence pour le cancer du côlon, le cancer du poumon et le sein, les maladies immunitaires, les maladies neurodégénératives, le diabète, ainsi que les maladies cardiovasculaires. Et ne pas oublier, bien entendu, les études de la populationnelle doivent disposer de notre génome ou de séquences de référence. Nous avons des insuffisances, on pourra en parler tout à l'heure, et nous avons des besoins. Il s'agit aujourd'hui d'aller renforcer nos capacités en matière de recherche-formation, de revoir notre organisation ou l'architecture de notre écosystème pour la médecine personnalisée. Il va falloir qu'on aille vers la numérisation, la digitalisation des datas, ainsi que l'analyse grâce à l'intelligence artificielle et de définir les infrastructures d'hébergement. L'accès aux tests, bien entendu, aux tests à des fins préventives beaucoup plus qu'à des fins thérapeutiques. De définir la collaboration au niveau régional et au niveau international, bien entendu, par rapport à nos problématiques et par rapport à nos centres d'intérêt, comme il s'est passé dernièrement avec l'Union africaine et l'Union européenne, lorsqu'il y a eu le workshop en Afrique du Sud, à Cap Town, où l'Algérie a été représentée par ce point focal. En conclusion, je dirais en synthèse, des initiatives ont été déjà lancées. Elles existent, mais les efforts sont vraiment dispersés et les niveaux d'avancement sont un petit peu hétérogènes, de sorte qu'elles ne soient plus ressenties d'une manière inadéquate par le citoyen algérien en matière de santé. Les besoins, nos besoins, mesdames et messieurs, les domaines d'intérêt ont été discutés. Ils ont été discutés lors d'un workshop qui a été organisé par l'Agence thématique de recherche en sciences de la santé en 2018, sous l'œil bienveillant de la Direction générale de la recherche scientifique et du développement technologique, qui a contribué à ouvrir deux voies essentielles et à de nouvelles actions en matière de médecine de précision et l'agriculture de précision. Nous avons une approche selon l'analyse SWOT qui va certainement guider nos pistes d'action à entreprendre en termes de priorité nationale, les thèmes sur lesquels on doit se concentrer, que doivent se concentrer nos chercheurs, nos praticiens et notre industrie pharmaceutique en fonction de nos compétences à l'échelle nationale. En outre, une action simultanée du lobbying au niveau national, une nouvelle structuration au niveau régional devrait contribuer à renforcer l'élan et à établir une nouvelle dynamique bien structurée. L'implantation, mesdames et messieurs, de la médecine personnalisée en Algérie apparaît imminente, nécessaire, vitale et indispensable. Je vous remercie de votre attention.
Pour professeur AOUFFEN, ça se voit que beaucoup a été déjà fait, a été déjà réfléchi d'une façon très intense. Et quand même, il y a toujours des questions qui en viennent un petit peu en tête quand on pense à la mise en place justement d'une plateforme aussi développée, mais aussi compliquée en Algérie. Et pour cela, j'ai pris quelques questions de la part de l'audience. Donc, il y a, je pense que c'est madame ou mademoiselle Emma Mardin. Elle pose une question, elle dit « Monsieur, est-ce qu'il y a un projet en cours de développement de databank d'ADN concernant les polymorphismes des maladies génétiques telles que le cancer et autres ?» Vous êtes, voilà.
Je suis sur le métier, effectivement. Les databanks, c'est en cours. C'est en cours parce qu'il y a un aspect éthique et un aspect juridique qui est en train de se formaliser, mais c'est en cours. Pas uniquement pour les maladies génétiques, mais pour le reste. La grande problématique, c'est la suivante, et je l'ai déjà dit dans notre approche d'état des lieux, c'est qu'il y a du travail qui se fait d'une façon hétérogène et pas homogène. Donc, chacun travaille dans son petit coin, en quelque sorte, et on n'arrive pas à rassembler, à mutualiser toutes les informations. Et assez souvent, il y a une duplication des informations, donc il est extrêmement difficile d'avoir pour le moment une réponse précise à cette question.
Est-ce que vous avez aussi pensé au stockage, justement? Comment vous allez stocker? Est-ce que c'est dans nitrogène, liquid nitrogen, ou bien dans des moins 80? Vous avez pensé à ça?
Oui, nous avons pensé à cela. Et, sincèrement, il y a plusieurs questions sur lesquelles on est en train de travailler, puisqu'on avait été mandaté sur cette question, et nous sommes en prévisionnel d'aller organiser un workshop pour la mise en place du réseau thématique. Et à partir de là, il ne s'agit pas d'aller sur notre vision est la suivante. C'est que nous avons fait une approche transversale de ce qui se passe en Algérie. Et on a remarqué que on ne fait pas dans le partage. Nous avons quatre points forts. 4 points forts, c'est que le gouvernement ou l'État algérien a financé des équipements qui sont très chers. Et il y a de la compétence, donc il y a de la ressource humaine. Il y a déjà des travaux de recherche qui sont publiés. Il y a aussi les financements, mais on n'arrive pas à les faire fructifier sur le plan pratique. Et là, on est en train de mettre en place une stratégie pour mettre le réseau national. Et comme ça, on définira une stratégie à l'échelle nationale et on pourra traiter tous les problèmes à l'échelle pas individuelle ou à l'échelle des laboratoires ou à l'échelle des unités, mais à l'échelle nationale.
C'est génial, c'est génial. Une autre question, est-ce que, concernant un petit peu aussi la recherche, est-ce qu'il y aura des équipes mixtes qui seront créées entre les hôpitaux et les universités ou bien les centres de recherche qui pourront justement faire bénéficier les deux en même temps, vous savez, genre les informations qui parviennent des équipes de recherche qui peuvent donner de nouvelles informations aux praticiens et vice-versa aussi, concernant ce qui se passe en Algérie.
Je vais me libérer. Depuis octobre 2022, notre ambassade algérienne qui se trouve à Bruxelles a saisi notre département ministériel pour aller sur une nouvelle vision incitative et immersive en matière de la collaboration à l'international en matière de médecine de précision. Et nous avons été chargés pour faire une réflexion dans ce sens. Pour rappel, déjà en 2018, le travail a été initié par l'agence. Nous avons organisé avec les Allemands, avec l'équipe de Francfort et de Max Planck, pour aller sur une nouvelle dynamique et une nouvelle vision de collaboration avec les Allemands sur la médecine de précision, et avant cela avec l'équipe chinoise pour tout ce qui est nanotechnologie et nanomédecine en matière de cancers avancés. La grande problématique qui a freiné un petit peu cette action, c'est que c'est en 2019, la fin de 2019, la COVID-19. Aujourd'hui, depuis 2020-2021, la fin de la COVID pratiquement, où les chiffres ont chuté, il y a eu un lancement des appels à projets, de programmes nationaux de recherche. Et là, on ne peut pas lancer un appel à projet tel que le programme national de recherche s'il n'y a pas d'équipe mixte. Et aussi, déjà en 2019, avant la COVID, il y a eu des lancements de 5 appels à projets, mixtes. Il y avait sur les technologies innovantes en santé, il y avait sur l'IA et l'intelligence artificielle et la santé, la recherche translationnelle en cancérologie, il y avait sur le scorpionisme, et sur le dernier point, c'est les technologies émergentes en santé. Voilà. Donc, pour répondre à votre question, maintenant, on ne peut pas lancer un programme juste pour lancer un financement pour un programme d'une équipe de recherche qui n'aura pas un impact sur la société. Donc, c'est lancer des appels à projets pour la création d'équipes mixtes, bien entendu.
Ok, parfait. On a une question aussi, celle-là elle est en anglais, donc je vais la lire en anglais. How about the personalized drug approval in our health system and their reimbursement by the insurance? Donc la CNAS et la CASNOS.
Si vous avez bien remarqué tout à l'heure, il ne s'agit pas. La grande problématique, c'est que les chercheurs en Algérie sont tous domiciliés, ils font de la recherche fondamentale. La recherche fondamentale, c'est à niveau d'une université. Ouais. L'une des visions de la Direction générale de la recherche, de notre département ministériel, c'est d'aller vers la recherche appliquée. Donc, comment cette recherche va faire bouger l'impact de la santé. Donc, c'est la recherche qui a un impact sur la santé. Assez souvent, nous avons plus de 1000 publications dans des grandes revues indexées à l'international en matière de médecine de précision sur les 5 dernières années. Mais on voit très peu son impact sur le terrain. Et dans ma présentation, dans cet état des lieux, de cette vision, de cette vision d'approche, on s'est rendu compte qu'il ne s'agit pas d'aller seul, l'enseignement supérieur. Donc, il s'agit d'une volonté d'un gouvernement, d'un État, et pas d'un département ministériel. C'est pour ça que j'ai cité le ministère du Travail et de l'Action sociale, le ministère des Finances, le ministère du Numérique, pour la mise en place des data et la protection de nos données, bien entendu. Et il s'agit de créer l'écosystème. Il ne s'agit pas d'une vision monoculaire de notre département ministériel de la recherche. Il va falloir qu'on inclue tout le gouvernement, et c'est pour cette raison, une des priorités aujourd'hui, c'est comment l'université pourra influencer la vie et comment elle peut influencer l'amélioration en matière de qualité des soins.
Donc finalement, l'université sera un petit peu la pointe de tout ce développement et que, comme vous avez dit, il faudra que toute une collaboration entre différents ministères du gouvernement puisse aider à la mise finalement à la mise en place de cette médecine personnalisée. Et une dernière question justement concernant cette collaboration qui sera mise en place à une échelle gouvernementale. Est-ce qu'il y a en fait un programme aussi avec les douanes algériennes pour créer un couloir rapide? Comment?
Les douanes.
Avec les douanes, oui, avec les douanes algériennes pour créer un couloir rapide afin de recevoir rapidement et sans perte de temps bien entendu les instruments, mais aussi et surtout les réactifs qui seront nécessaires pour conduire la médecine personnalisée en Algérie. Parce qu'on sait très bien que un des points difficiles à surmonter, même dans la recherche, dans l'université, pour la recherche fondamentale, c'est ce problème de perte de temps au niveau des douanes quand on veut ramener donc des réactifs, que des fois, c'est en fait des simples réactifs. Et donc, est-ce que cela a aussi été pris en considération pour pouvoir justement avoir une médecine de précision efficace et de pointe, bien sûr?
Je crois que j'ai déjà répondu, M. NAIT Mohamed, à la question qu'il s'agit, oui, entre autres. La médecine personnalisée, c'est une ombrelle en quelque sorte. C'est une ombrelle où il faut la collaboration de tous les secteurs. C'est pour ça que la recherche seule, ou l'innovation seule, ou la vision seule du ministère de la Santé, il faut que ce soit un problème partagé par tous les secteurs, tels que les finances et les douanes, bien entendu. Donc, si on doit avoir des équipements, il faut avoir des financements, il faut avoir des couloirs verts pour les tests, pour les équipements, etc. Donc, il s'agit de prioriser un petit peu la vision des choses. Moi, je voudrais revenir sur un point qui me paraît essentiel. Ce workshop pourra ce workshop pour la mise en place du réseau thématique de la médecine personnalisée qu'on essaiera de mettre en place et de l'organiser assez rapidement avec le soutien de notre département de tutelle aura à régler deux choses. C'est la mise en place d'une société savante pour la médecine personnalisée et c'est de là qu'on pourra faire avancer les choses puisqu'une société savante aura plus de facilité pour l'approche transsectorielle. Parce qu'il est difficile de passer d'un secteur à un autre sur le plan administratif et sur le plan relationnel, peut-être parfois. Et la deuxième des choses, c'est la rédaction du livre blanc de la médecine personnalisée. Les choses prennent du temps, mais elles se font en profondeur pour régler de façon pédagogique et hiérarchiser un petit peu les priorités. Si vous reprenez un petit peu le tableau SWOT que j'avais présenté, vous allez voir qu'il y a un élément sur des menaces, c'est le manque de communication intersectorielle qui pose réellement des problèmes dans le développement de la médecine de précision, entre autres, mais pas le seul.
Merci beaucoup. Merci beaucoup encore une fois.
Merci à vous.
Merci دكتور أوفن. وكما يقولوا good things happen fast يعني just بالخف because we are so enjoying it. يعطيكم الصحة على كل المشاركين les volontaires اللي مدوا من وقتهم وخاصة في شهر رمضان باش يورغانيزيو هاد la conférence للاحتفال بيوم العلم. Some of them on short notice ومالغري ça they showed up. يعطيكم الصحة وكما يقولوا إن شاء الله نتلاقاو في محاضرات أخرى إن شاء الله. I encourage you to keep in touch. قعدوا ما يعني keep in touch and keep helping كل واحد باش يقدر وكما يقولوا كل حاجة عندها الزكاة تاعها وهذه زكاة العلم إن شاء الله. وبهذا يعطيكم الصحة لكل المشاركين كل المشاهدين. وإذا سجلتوا في هذه المحاضرة راح نبعث لكم certificate of attendance و même temps نبعث لكم لي ليان باش تقدروا تشوفوهم ولا you can share them يعني اللي بارطاجي مع لو ريزو تاعكم إن شاء الله. وشكرا جزيلا ونشوفكم في أقرب وقت إن شاء الله. السلام عليكم.